In the previous videos, you have seen how a seed goes all the way to a plant. So you must have been wondering how a tiny seed with DNA packed inside can grow into a big plant like this. So this is where the wonders of cell division comes in place. So the cell division. So let's take a closer look at what is a cell. So cell is nothing but basic unit of life. Basic unit of life. So if you take a cell, so most of the cells are represented as a circle, and we have a nucleus in the center, and we have DNA in the nucleus. So this outer membrane separates the cell from the external world. So it protects the DNA inside the nuclear envelope, inside nucleus. So this is cell. So how did a small seed grow into a big plant? How did a small seed grow into a big plant? So if you talk about a small cell growing into an organism that is trillions and that has trillions and trillions of cells, so we need to talk about something called cell division. Cell division. So what is a cell division? Cell division is nothing but there will be one parent cell with DNA, and it divides itself to form two daughter cells, and gives its DNA to the two daughter cells. This is cell division. So if you come to the cell division, so there are two types of cell divisions. Mitotic cell division or mitosis and meiosis. So for mitosis, body cells undergo cell division. For meiosis, sexual reproductive cells undergo cell division. Let's take a closer look at mitosis. So mitosis has phases in it. So if you take a cell and record what's going on inside a cell from where the parent cell is getting ready to divide until the daughter cells are formed, you can separate them into phases as 1, 2, 3, 4. So we will be talking about these phases now. So mitosis or mitotic or mitosis phases. So first phase is prophase. Second one is metaphase. Next we have anaphase. And next we have telophase. Let's take a close look at prophase. So when talking about prophase, we need to remember the phrase DNA condensed or condensed DNA or condensed DNA. So what's a condensed DNA? If you take a DNA in a normal cell, it's going to be like that. And if you take a cell that is ready to divide and it's and it's in its prophase, then it's going to wind itself up into a structure like that and into a structure like that. And this is one chromosome. And in prophase, it duplicates itself into two chromosomes. Now let's take a look at the cell. So this is a cell, and we have nucleus, and we have DNA that's been wrapped around, DNA that's been wrapped around itself to form structures like this. And we have centromeres that attach the two strands of DNA. The chromatids, these are called chromatids. Okay, let's write it down. These are chromatids, and these are centromeres. Centromere. And in prophase, we also have something called centrosomes that duplicates itself and to form another centrosome. And you'll see in the next step. So the first phase was prophase. Next, we'll go on to learn about metaphase. So before going to the metaphase, we have something called pre-metaphase. Pre-metaphase. So in this phase, we have a cell. We are talking about pre-metaphase now, and the nuclear envelope dissociates itself. It fragments it. These are the fragments of nuclear envelope. And what happens to the DNA? DNA as the fragments, as it fragments, and DNA is let go free now. Let go free now. So these are the centromeres, and this is pre-metaphase. And let's see what happens in metaphase. In metaphase, there is no nuclear envelope to hold the DNA. So what happens is we have the chromatids line up in the center, in the midline of the, this is the midline, this is the midline, this is the midline. We have chromatids, and we have centrosomes that pass the microspindle tubules. Okay, microspindle tubules. Okay, that pass attached to the centromeres, and these will put the chromatids in position. This will put the chromatids in position so that they can be pulled apart. So this part of chromosome will be pulled towards this side, and this part will be pulled towards this side. So in the next phase, which is anaphase, we can see that we have a cell, and the chromatids are not attached to each other anymore. The chromatids are split. They are being pulled by the centromeres towards themselves. They're pulling towards the centrosomes. The chromatids are being pulled. So the next phase, we have telophase. Telophase, what happens is, so the cell separates to form two daughter cells. So the cell elongates, and the nuclear envelope begins to form, and the centrosomes move towards the corner, and the chromosomes, these chromatids, now they can be called as chromosomes. They get equal amount of chromosomes into each cell, and there is a split in the cytoplasm of the cell. So there's a liquid, and in the outside of the nucleus, outside of the pink line, that cytoplasm gets split at the center, and these cells will be divided into two cells. These divided into two cells with completely formed nuclear envelope and DNA inside. So two daughter cells are formed by the end of telophase. This split is called cytokinesis. So in mitosis, We have a parent cell with two chromosomes, and it divides to form two daughters. It divides to form two daughter cells. So daughter cell 1 and daughter cell 2. Each daughter cell will have two chromosomes just like the parent. So the number of chromosomes do not change. So this is one of the important note. So in mitosis, the daughter cells will have the same number of chromosomes as the parent cell. Let's take a quick review of what we have done. So we went through prophase, where the DNA condenses, metaphase, where the chromatids line up, anaphase, where the chromatids separate, telophase, where the cell splits to form into two daughter cells, and cytokinesis, where the cytoplasm splits to form two daughter cells. So this is the process where one parent cell will be divided into two daughter cells. I think if you have liked my video, please like it and share it with your family and friends. And if you want more videos like this, or if you want anything else that you want to learn, please let me know in the comment section and I'll make a video for you. Thank you so much for watching and have a good day.